Hi, we're going to be starting Chapter 18 today. We'll be doing the first half on microevolution. And as always, I want to remind you to take notes during the video and especially jot down any questions that you have so we can talk about those during class. These topics on evolution are a little bit unusual here and there, some things maybe we haven't thought about before. Definitely bring your questions into class so we can talk about them. Now, when we're talking about evolution, we need to remember that we are discussing populations. Only populations can evolve. Sometimes we say species evolve. Species and population can be used kind of interchangeably in this case. You yourself are never going to evolve. An individual does not evolve. So when we're talking about evolution, it's a change in a population over time. And in order for things to evolve, we need to have variation. We talked about that back in Chapter 17. So today I just want to start with reminding you what some of those sources of variation are. And gene mutations are absolutely a key source of variation, a way to get some different DNA sequences from what you had before. You can also have changes in either chromosome number or structure of chromosomes with a chromosomal mutation or a non-disjunction during meiosis, change in the number of chromosomes. That give you some variation. During meiosis 1, as the paternal and maternal chromosomes are getting all separated and sorted out, that's going to give you lots of variation, of course. Also during meiosis 1, there's crossing over between those non-homologous chromatids, the non-sister chromatids within a homologous pair. That crossing over will give you even more variation. And of course, when you mix your gametes with someone else's gametes, that fertilization will give you a new set of gene combinations that's never existed before. All of these are all sorts of rich sources of variation. However, of all those, only the mutations will create new types of genes. Only a new gene mutation will create a gene that didn't exist before. So those gene mutations end up being really important as far as variation goes. And just a reminder that not all variations are good, and not all mutations are good. Some of them can be good, some can be bad. Probably most of them have no effect whatsoever. Most of them are probably neutral, and we'll talk more about that in a little while. Now, one really key concept in evolution is the idea of genetic equilibrium. If you remember, the term equilibrium refers to something being kind of at a steady state. Well, we're taking a look at the genes in a population when we discuss genetic equilibrium. If a population is evolving, that means the population is changing. The only way for the population to change is for certain traits to become more common or less common. The only way for traits to become more or less common is if alleles are becoming more or less common, since we're talking about genetic traits here. And as some alleles become more common, we're going to kind of assume with evolution they're becoming more common because they are something that are beneficial and help you to either survive or reproduce. Alleles become less common if they keep you from surviving or keep you from reproducing. Well, a couple scientists whose last names were Hardy and Weinberg did research on this, and so we refer to this as the Hardy-Weinberg rule when we kind of look at allele frequencies and when they change and when they don't change. And one of the key ideas with Hardy-Weinberg is how you could possibly get no evolution. How could you get no change in allele frequencies? And they came up with a list of criteria, and in that list we have, first of all, no mutations, because a mutation would change allele frequencies by definition. So you'd have to have no mutations. You'd have to have a very, very large population. If you have a small population, just random chance could bring about change in allele frequencies. So you've got to have a large group to kind of buffer out those random changes. The population has to be isolated. If individuals are moving in or moving out of that population, that's going to change allele frequencies also. Everybody has to survive and reproduce, because if not everybody survives and reproduces, then that means some people aren't passing on their alleles, and that's going to change frequencies. And mating has to be random. In other words, you wouldn't be picking and choosing who you would be reproducing with. Each reproduction would just be simply a random case. And if you have all of those factors all matched up and all truly the way it says here, you'd end up with no evolution. So if all those conditions are true, the population would be at genetic equilibrium. Now, each one of those is kind of a weird situation. To picture no mutations, to picture a fully isolated population, They're all a little bit strange. We can picture how they could happen, but in terms of all of them happening, we think that really doesn't happen. We try to cover our bases here and say that it's almost never all true for a population, so we say that populations would almost always be evolving. The truth is we think they probably always are. Now you're going to hear Hardy and Weinberg's names in class a little bit more as we do some mathematical analysis of the allele frequencies in a population. We're going to use the Hardy and Weinberg formulas. We're going to be using some algebra. We'll be using terms p squared, 2pq, and q squared. That's something we'll be doing in class because it's going to take some practice to get used to how those problems work. Now, another key idea in evolution in a population is the idea of genetic drift. Now, if you hear the word drift, it's supposed to kind of make you picture something like if you have a river flowing along and there's a raft on this just kind of drifting along down the river. That raft isn't controlled, nothing is guiding it, it's just simply being carried along by random currents. And that's the idea of genetic drift. This is a random thing we're looking at here. This is not the same as natural selection. However, genetic drift does lead to evolution also. Genetic drift is when we have random changes in allele frequencies. We call them fluctuations because they're not moving toward any specific direction. It's not like a certain trait is becoming more common for a reason. It's just over time, because of random things, a certain allele may become more or less common just due to weird events that happen. And genetic drift is highly exaggerated in a small population. If you picture, for example, if you've got a group of 20 people and one of those 20 people just falls off a cliff accidentally, falls off a cliff and dies, and of course wipes out all their alleles. Their alleles aren't going to be in the population anymore. In that population now of 19 people, there's going to be a pretty big shift in allele frequencies just by the loss of one person. Now, if you have a population of 5 billion people and you lose one person, that's not really going to change allele frequencies noticeably. The larger population is, the more it can absorb these drifts that happen. But in a small population, they become very noticeable. Now, another idea in genetic drift is something we call the founder effect. And the founder effect is when you've got a large population in one area, and a small group of that population goes off and founds a new population. It becomes a new population with its own new allele frequency. By the way, we're using the term allele frequency here. In everyday language, it's often called the gene pool, but in science, we often kind of shy away from the term gene pool when we're discussing evolution because it doesn't give us the mathematical analysis we really need. But again, picture a large gene pool. We're now going to take a group out of there, and they're establishing a new gene pool. The allele frequencies in that new founded population are going to be different than the original just based on who left. And if you picture something like colonists from Europe traveling across and moving to the Americas, they would have created a new population with a new allele frequency, and that'd have some genetic drift to it. Another type of genetic drift that's very closely related to the founder effect is something called a bottleneck. And a bottleneck is when something really drastic happens to a population and most of the members of the population are killed off. And this could be like a natural disaster, a massive war, a disease, a plague going through. If you reduce almost all of the members of the population down to just a very small amount, that small group is going to have a new allele frequency compared to the original larger group. It's similar to the founder effect. The difference is in the founder effect, you've got the large population, the small group leaves, and now there are two populations. In the bottleneck, you have the small group, but the larger overall group is gone. They're dead. So we have the same concept, but two very different ways that this takes place. Now, the term founder effect, obviously, it's picture like the founding fathers coming across from Europe and starting a new population here in the Americas. The term bottleneck actually refers to the actual neck of a bottle. If we picture something like here, we have a bottle with a bunch of little marbles in it, and the marbles represent different allele frequencies. We can see, just looking in here, we have some kind of greenish ones, some purplish ones, and some kind of orange ones. And each one has its own particular frequency. And as this bottle gets turned upside down, because the neck is small, only a few get out, and then the rest of the bottle is disposed of, and we keep just the ones that come out of this bottlenecking event. In the new population, there are different frequencies. In this case, we eliminated one allele, The green one became exceedingly rare, and almost the entire population is that purple type of allele. So this bottlenecking event can greatly reduce what you have in terms of your allele frequencies just due to chance events. It's not that the orange marbles in here were bad. It's not like they were unable to reproduce. They just happened to not pass out through the bottlenecking event. So when we discuss genetic drift, as we said, it leads to evolution. It is a way to change the allele frequencies, and specifically what genetic drift does is it reduces variation. Genetic drift won't increase the variation because it's just random events kind of wiping out certain alleles from that population. And not only does it reduce the genetic variation, it reduces it in a random method. So the new population, the new allele frequencies have different percentages to them, but it's not necessarily good or bad for the population. It's just different. Now, one thing that ends up happening anytime you reduce variation, you leave the remaining population more susceptible to a change. Let's say we have a bottlenecking event with humans on Earth, and we somehow wipe out, let's say, 6.5 billion people. We're left with about 500 million people on Earth. Well, after a bottlenecking event with that 500 million people, there may be another event, something like a disease that comes through that wipes every surviving human out. Something that maybe would have left some survivors if we'd had all the original 7 billion plus humans on Earth at the time. By wiping out, by random chance, you could eliminate some people that might have survived a future environmental change. So genetic drifts can be very important in terms of the future of a population. That's the end of the first part of Chapter 18 for us here. And again, bring your questions to class. And in the second half of Chapter 18, we're going to discuss some other ideas of things that happen with populations. Have a good day.